And we are done with the mollusks. So this section is all about mollusk genetics. And so we're going to look at a couple different ones. This one is from Crassostria gigas, which is a Pacific oyster. And some of the other things we're going to see down there are going to be cephalopod genomes. And so, pardon me, for Crassostria and for the oysters, they are actually going to have 19 chromosomes. So you can see all the little chromosomes there. And they actually have only a few genes in common, about 9,300 in common. And all the rest are going to be specific to one or more groups or specific to one or more individuals. So even though we classify all these things as being in the same group, these are all bivalves, they're all in the same group, which is the oyster-like bivalves. They're still pretty different. Compared to each other. Far more different than comparing you to a chimpanzee or even you to a mouse, I would say. And let's start this up and get that terrible transcription going. There's our subtitles. Okay. So here is a little plot comparing sort of the order in which genes are found on the different chromosomes between oysters and some other creatures. And so here we're comparing bilaterans, so things that have bilateral symmetry. And here's the group. And so if they line up with the same things in the same places as in mollusks, then you're going to get a diagonal running down the middle. So we're comparing one mollusk to another. And this is a, this is a scallop, and you can see Pectin yesoensis. You can see it makes a nice little line there, and so the pectin is going to be similar to a scallop, but or it's similar to that particular kind of oyster. But compare the other kinds of oyster here, and you see that their genomes are just completely reorganized. Not only do they have new genes, but the genes are chopped and reorganized in interesting ways. And compare those to S. mansoni, and it's just, nonsense. This would be an annelid. So there's basically no real comparison there. Compare it to a fruit fly or Cinerabdidus elegans, which is a nematode, or compare it to a flower beetle or a Anopheles mosquito, and you can see it's just chaos there. But compare the order of genes to something like us, a Homo sapiens, and bam, they pop back into line. So we've got a kind of a molluscan order to our genome, and we've got nice matches for a lot of things that you find in mollusks. So that's kind of neat. And we're going to be similar. Mouse, chicken, and Danio are the little fish that we have in 210, UC 210, the zebra Danio fish. Okay, so mollusks reorganize their genomes quite a lot, like quite a lot more than any other group. For some reason, their genome is just able to be recombined in many interesting new ways. And when this happens in humans, it usually means either cancer or death. But when it happens in mollusks, they just seem to roll with it, which is pretty cool. So let's look at their Hox and Parahox clusters, shall we? So here we've got our actual annelid down here. And this is a lingula, so this is going to be another one of those things, a brachiopod, but it's going to be another one in the that has trochophore larvae, the trochozoa. And then here we're going to have all legitimate mollusks. So we've got our cephalopod, and we've got a bunch of other mollusks, mostly bivalves, I think, down here. And you can see here we've got our Parahox cluster, and so the first one of those Parahox genes all was conserved, but you can see that some of these things move around a little bit. And but there's the Hox genes coming in. So Hox 1 through 5, these are going to control where different parts of the body are going to grow. And then you've got Lox genes, which are going to be similar to the Hox genes, and the Post genes, which are also going to have similar functions. These are actually expressed in roughly the same order, even though they are rearranged very thoroughly. But the way they're arranged in the genome normally correlates to the order in which you find them expressed. And so they'll be turned on in clusters, and like LOX5 will work first, then ANTP, then LOX4, then LOX2, something like that. They also tend to be clustered in order of where they appear on the animal. So things that form the head of the animal will be in one place, and things that form the tail of the animal will be in the other place. And so here you can see these are the zones of early Hox gene expression in a chordate embryo. This would be something like a human or something like a chicken. And so you can see our Hox5 cluster is going to form the top part of the spine. Then Hox6 is going to form the next part. Hox9 is going to form that whole chunk. Hox10, this chunk, and helps form the hind limbs. And Hox11 is going to form everything back here, the caudal and sacral vertebrae. But then you get some of these other guys. So take, for example, one of those, let's see, scallops, so fairly sensible, the way they're programmed. Oysters, fairly sensible. Bristleworm, an annelid, fairly sensible, fairly similar to what we see there. Compare that to an ascidian, which is a pretty close relative of us. This is one of those primitive chordates, or a mouse, and you can see the same general order. And so the mouse is going to express these all in one cluster, expressing them in this sort of way. And what we see with the mollusks is that they tend to express them in groups. So they'll have different groups that can be turned on as a group rather than having to turn on the entire program at one time, which is the way it is kind of done in mice. So mice are a little more organized. They've got their genes together a little bit better. And the fact that each of these little clusters can be turned on separately means that if they happen to move to a different chromosome, if they end up out of order, it doesn't really matter, because they can each be turned on separately at the right time. And so, yeah, you're going to make a good-looking molluscan body. All right, so let's also look at the PAX genes. So some of these PAX genes are actually going to turn on the opsin genes. And so this is a couple of things that you know about coming together. PAX genes are going to be the marker of the parahoxozoa, which is pretty much everything, right? Except for just a couple groups. And yeah, in each of these different organisms, you have different types of opsins. So in humans, we actually only have C-type opsins. Now we have 3 different versions of the C-type opsin for seeing red, green, and blue light. And we have a 4th version, which we use to see just light in general. This is the one in the rods. But genetically, we do not have any greater diversity than that. We only have C-type opsins. We have lost all the other ones. But look at scallops. Look at their eyes. These are going to have C-type opsins, like us, and G0-type opsins, and R-type opsins, which is like the original rhodopsin. And they're going to have multiple PAX genes that turn these things on, whereas we just have PAX6 that turns on our one, lonely C-type opsin, as in this little cephalachordate up here, which is going to be a relative of ours, a thing called Amphioxus. So looking at the anatomy, now if we were in class, we would be dissecting these nice big fat squids. And they are so cool to dissect. Yeah, I really hope there's a chance that we can do this. But inside you'll see they have an ink sac right down there. Boop. And the shell, if they still have a shell, is going to be protecting this ink sac. That's kind of where the shell goes in the adults. They start out as a shelled larva usually, and then they'll absorb the shell as they grow. And in something like a cuttlefish, it'll form a little plate that'll also protect that ink sac. And then the rest of them is a great big siphon, and they have a beak that looks a lot like a parrot's beak right in here for actually biting off chunks of food. These 2 extensible tentacles will actually stretch out like, like rubber bands. And they have suction cups on the end, and they will grab the food and whip it in toward these little tentacles. They're the arms right around the mouth. And then they poke everything in so that little beak can tear up whatever the food is and swallow it. Yeah, so that's how their eating works. And this is a chambered Nautilus. It's the same kind of thing. So it's going to move hydraulically. It's going to breathe in water here and it's going to spit water out with this funnel. And so it's always going to be moving backwards. And so when you see these things in the wild, there are times when their shell does crash into things because their eyes are on the side. They can't really see where they're going. They can just see what they're trying to eat. And so I guess it could pull in water from here or spit it out, depending which direction it wanted to move, perhaps. Okay, last couple of little bits of the genetics here. So cephalopods have a thing called RNA editing, which is kind of neat. And so there is a adenosine deaminase would actually convert an adenosine to a different nucleotide. And so some creatures, including humans, will actually do this. So this is a way that we can make one gene and then we can go back in and edit that gene and we can actually turn it into different versions of the gene. We can make it stop, or we can make it continue and read a little bit extra sometimes, or we can go in and change what one amino acid is going to be on the protein that's encoded by the gene. And we do this with ADARs, these adenosine deaminases, and that act on RNA, ADAR. And we think these are a molluscan invention that we may have stolen or may be present in the last common ancestor of us and mollusks. And so this is what an adenosine deaminase does. It takes an adenosine, which looks like this, and it pops off the amine group up here. So this NH2 is going to add some two other H pluses from a water molecule, and it's going to take that oxygen from the water molecule and attach it on here. So it's going to do that little piece of chemistry. And it's going to convert an adenosine into a thing called an inosine. The interesting thing is inosine will actually, it's a nucleotide, but it's not one of the four that we normally use, but it does base pair with any of those four. So if you have an inosine in there and you go back and try to read it, then the inosine can encode for anything. So that's kind of neat. And this is the ADAR protein structure solved in conjunction with what looks like double-stranded RNA here that it's presumably going to be editing. So there it is, reaching its little protein tentacles down there and editing the RNA. Okay, and so oysters, fairly sensible. The way they're organized, they've got their Hox genes in little packets, and that's great. And in humans, we tend to have one big packet, but we have that entire packet represented 4 different times because we actually have 4 copies of everything in our genome. It's just that we've lost certain parts of this. So we think that the human genome along the way from primitive genomes to ours is actually duplicated twice. And you can find animals that have 1 or 2 copies or all 4 copies of basically all the genes that we have from these duplication events. And they're all different kinds of what we would call fish, but they're kind of weird primitive fish that have these. And so branchiostoma is at the bottom of that branch. This is one of those cephalochordates, one of those really primitive things that starts out looking like a human embryo and just stays that way for all of its life. And so you see it's got the same genes, but it only has 1 copy in order. And then there are other things halfway up that have 2 copies of everything that we have. So anyway, Drosophila melanogaster, this is the fruit fly. Cetalita, I don't remember. Actually, no, both of these are mollusks. These are going to be those other bivalve mollusks, I believe. And you can see they've got things. They're organized in little clusters. The clusters can be in different places, but the cluster has to remain together. Then look at the octopus. It's taken this to the next level. So it's got most of the same Hox genes, but instead of grouping them up all in one group like our ancestors did, or breaking them into separate groups so we could move them around to different chromosomes like the other mollusks did, these guys have split up their Hox genes so that they are nowhere near each other, and each one runs independently. They're not going to be activated in groups. So that means they need more and more complicated other transcription factors to turn these on, but you know what? It still works. So when the octopus genome was first sequenced, the people that sequenced it actually remarked, they said that there's no way octopus can be related to the other mollusks. And they suggested that it may have extraterrestrial DNA in it. That is, of course, silly. And if you actually make a phylogenetic tree that compares these things, you'll see the octopus group right with all the other mollusks. They have the same genes. They're just extremely scrambled, the order of the genes. And so that's unusual. They just seem to be able to move their genes around a little bit more than any other kind of life that we know about outside of maybe bacteria. And maybe even more than some bacteria would like to do this. So yeah, octopus, very strange, but they are a cephalopod. And shown here, cephalopods have a lot of things that are going to be similar to the genes that we have in humans. So interleukin 17A is something that we thought was only in humans, and yet they find one in an octopus genome. Pardon me. This is going to be involved in controlling the immune system. All these interleukins are little proteins that cells of the immune system share between each other. They're little signals that tell one when to start or stop or when to call in reinforcements. And sure enough, we have them, and these mollusks have them as well. So that was just a little bit on molluscan genetics, because I think it's an interesting topic. And we have one last little section here, and then we're done with the mollusks.